Let me now introduce you to the Turing-Roche Knowledge Share series. Its aim is to bring together members of the Turing-Roche networks to the wider scientific community to hear different perspectives on topics in data science and build new connections. To get us started, let me go through a couple of slides to give you an intro of the partnership. Oops. As per the name, it is a partnership between the Alan Turing Institute and Roche. I guess most of you are familiar with at least one of these organizations. In case you're not, the Turing Institute is the UK's National Institute for Data Science and AI, and it's built around a network of university, industry, charity, government partners. Roche is a leading healthcare company with a long-term interest in personalized healthcare, and we're one of the growing number of academic partnerships. The partnership was launched in 2021, and its 5-year mission is summarized by our partnership North Star. The goal is to collaborate to achieve a better understanding of patient and disease heterogeneity, essentially why patients respond differently to treatment and why diseases can manifest in different ways in different people. Our approach is illustrated on the right, with our main focus being on developing methodologies in statistics and machine learning to improve healthcare. We have focused our research on 3 different things: structured missingness, to deal with missing data, in particular when it exhibits in a structure; predictive modeling, to construct models that are robust to uncertainty and heterogeneity, and that are transparent; and multimodal analysis, to effectively combine different data modalities to improve clinical care. Our partnership core team is pictured here with representatives from Turing and Roche, which is expanded by our research associates, funded by the partnership and the enrichment students. And without further ado, let me allow Dr. Deepak Parasar to introduce the speaker for today's event.
Thanks, Maria. It's a great pleasure to have Dr. Veera Baladandayuthapani to present our knowledge share series for the Turing-Roche partnership. So Veera holds the Jeremy Taylor Collegiate Professorship at the School of Public Health at University of Michigan in Ann Arbor, and he's also the chair of the Department of Biostatistics, as well as the director of the Cancer Data Science Shared Resource Center at Michigan. And a lot of what Veera has been doing is kind of world expert in Bayesian probabilistic modeling and machine learning methods applied across a broad range of problems in medical and health sciences. And specifically a lot of work in multiomics, large complex data sets. And today we'll be hearing from you about spatial heterogeneity in the tumor microenvironment using an advanced statistical and AI machinery. So a very warm welcome. Thank you. Go ahead and share my screen. Let's see. That good? Yeah.
So, thank you, Deepak, for inviting me here, and of course, Maria for organizing this. A little bit of a fanboy moment, coming to Alan Turing Institute, and hearing so much about it, and thank you for allowing me to do my spiel here a little bit. So I want to make sure, can you all hear me fine here? Mm-hmm. This should be okay on Zoom as well. If I need to mic up, I'm happy to. So, and thank you to Turing and Roche as well, for allowing me this platform. So I want to talk about a few like Deepak mentioned, a class of modeling problems and actually scientific context-driven modeling problems in spatial biology that have gotten me very interested in and giving you sort of a sense of flavor, of especially in the cancer context about some unique modeling problems as well as their the contextual aspects of what motivates that problem. So I'm a statistician by training, and I apply as a biostatistician by trade now, and more specifically a cancer biostatistician. And something I've sort of learned along the way and learned to respect is biology can happen at very, very different scales. I know there's on the one end of the spectrum is the physiology aspects of it. I'm going to use my cursor here for those on Zoom, and then most of the actions sort of happen within the cell. That's where the genomic components live, and things get disrupted. To give you an idea about the scale that we are talking about, we're talking about like even cellular, subcellular levels. So it even goes down to like tiny microns, and that's where So and this is a book actually I started reading for fun. I don't know, I'm going to Sid Mukherjee. He has a series of 3 books. The first one is The History of Cancer, the Chief, this one. And it ended up, this book, I read it, now it became like a workbook, and for work and give you a lot of insights into why the cells are so important, what's the next paradigm in cellular therapies and such, and just contextualizes broadly the work, especially the quantitative work. As I mentioned, like cancer has been my scientific identity for a good part of 25 years now, starting even with my PhD dissertation. So I know a bit about the disease, not by no means an expert. Specifically, just the context surrounding this, the tumor microenvironment that I mentioned in the abstract. So just to give you a very high 70,000 foot, even 100,000 foot view, So basically, it's an ecosystem that surrounds the cell. So a tumor is a living, breathing, collection of rogue cells that's in the body, and it consists of many different types of cells. And actually, I forgot to mention one thing. So I have a tiny URL here and a QR code, so you can scan this if you want to just follow along with the talk. And I'm happy to, you can. These slides will be public, and you can, take a look at them later too. So it's tinyurl/TuringVB, and that's the QR code. All right. So it consists of many different types of cells, and each cell has its own function. So there's the immune cells, there's cancer cells, there's different cells, and these are the types of cells. You can see your different names here. And there is always a constant interaction, and especially when the tumor lives in a human, there's a constant interaction. If you actually click that link, I'm not going to click it right now, but you can actually see a nice simulated figure of how what is called a natural killer cells interacts with the tumor cells and kills it, actually, blasts it out. So it can be both positive and negative. Positive means it comes together, and negative means no, sort of part of our geography. And quantification of this is especially relevant, and I'll show you a few examples to make it a bit more concrete, but especially in immunotherapy, which has sort of been a paradigm shifting sort of class of treatments, interventions in cancer that's come about over the last, say, 10, 15 years or so. Which basically the underlying gist of it is, let your body fight against cancer as best as possible. So it works in some cancers, it does not work in some cancers, even which work, it does not work for the majority of the patients, So understanding, so this whole talk is based on like the more discovery science and how to understand that. And that's where the whole spatial aspects break in, that the technology is maturing so much over the last 5 years, especially in the last 2 years, allows the systematic assessment of this tumor microenvironment, which is standing list. So I want to just walk you through like what is the types of data here, and give a little bit of a context. I know I'm speaking like a lot of this before so that you get the why of it, and before I get to the how of it, So bulk genomics, which has been around 20, 30 years, like the standard gene extraction, You can think about like it's a collection of cells, you draw some biological materials of the cells, you put it through a machine, right? And then you collect, it can be DNA level data, it can be mRNA level data, right? So what happens is that you're getting a mixture of cells. It's a gimish. So you can think about it like a smoothie, right? You can't parse it out. So technology matured, last say 10 years or so, especially last 5 years, single cell technology is very good parse out each cell. So there was a specific technology that can isolate each cell, and then now you can measure the genomic components of each of the cells. So you can into sign, okay, what were the components of this? The spatial aspect of this, I can think about it like a fruit. Sorry for the fruit analogy here, but it makes it more intuitive here. Now you know exactly where they are placed. So you know the specific organization because each cell, you have barcoded it, and that's what the technology does, right? So this has been around like in various forms, spatial biology. It's one of the earliest ones is just plain histopathology, which is basically the pathology images that a pathologist sees to stage the disease, right? So these are whole slide imaging of the tumors. When somebody gets a biopsy, it gets put on a chip and stained by specific type of dye or with H&E, for the years in, and so that's your morphology. More disrupted morphologies, the higher stage of the gene. So that's what spatial proteomics actually are built up on the same kind of gist of that technology, but it's basically measuring proteins and tells you about the functional state of the cells. And spatial transcriptomics is actually one level higher, which tells you about gene expression. So each cells give you a gene expression. And then, spatial genomics in general is, getting multiple types of genomics here, spatial aspects of it. And now, if you put omics in it, it can be any kind of omics, metabolomics, micro, even multi-omics, you can get, there are some technologies that are emerging and data that's emerging that you can measure both RNA and protein on the same cell. And as usual, this has received a lot of interest from the scientific community. And as you can see, at least the last 2-3 years, the Nature Medicine has highlighted both spatial proteomics and genomics and transcriptomics. So just to give you an idea, like as quantitative folks, like what is the actual sort of data that one loads up in a computer and manipulates, right? So the pathology imaging aspects is, I know there's some experts in this room. The digital pathology is, looking at these images and extracting some kind of features, right? They can extract a lot of features from this, these H&E images, and tells you about the morphology and the tissue structures, right? And some are visible to the human eye, but some are not. So can you extract, So these are routinely collected as part of the staging and the diagnosis of cancers. Can you extract something, more from it? So there are many different types of features. One specific is one can actually infer the cell types from research images. So just think about simple 2 types of cells, right? Cancer and immune, right? So you can infer them from this. And now the cells, what the identity of the cells is, and then the spatial location. And these are images. So basically it's taking pictures. So these are scalable. Pathology, every patient gets it. So, data sets can range into hundreds, thousands, even tens of thousands for that matter. And since cancer is a genetic disease, some of these patients might have associated genomics data, genomics data collected on them. And of course, there's outcome level data, how did they respond to treatment, survival, etc. So the multiplex imaging is, as I said, it's a variation on that technology, histochemistry-based approaches, where the multiplex is actually the exact same technology, but now instead of just one dye, you put multiple types of dye. And these dyes are basically antibodies for different proteins. Think about, we have a slide of the biopsy of the tumor, you paint it with different colors and keep taking pictures. So the different colors correspond to different types of cells. So that's the multiplex nature of it. So you can simultaneously quantify instead of inferring cell types, you actually know the identity of the cell type. So each cell gives you, is it a type of a T cell, the type of a B cell, because they have different functions, that they play. So the structure is, again, you have the spatial locations. The modern technologies have grown up to assess more than 30, 40 types of cells. Even actually, I have a data set that we are working with, which is 60 types of cell types. And then some are phenotypic. What phenotypic means type, what is the identity of the cell, and the functional is what is, proteins are functional so that they control all the body functions in the body. So again, it's imaging based, so scalable. It's taking pictures and it's scalable across patients as well. So just to set it apart from transcriptomics, there are imaging based transcriptomics too, but in the case of transcriptomics, it's the exact same sort of flavor of the data or the structure of the data, but just now think about that from each of these location, which could be either a single cell, that's where some technologies, and some are a collection of cells, maybe a few cells, 5, 10 cells. And then from each of these, think about like a 20,000 dimensional vector, like that's the number of coded genes. So that gives you a deep dive into the mRNA expression, the programs of it. So it's kind of like high N, you have lots of cells, but also you have high P, which is a lots of genes. No, but this is sequencing based. That means there is a lot more technical aspects to this that it has to go through. It has to be for sequencing platform. So it's not really what is called scalable. So the number of patients that you do, that is the number of cells that you get. Biological replicates are still low. You can get deep dive into the mechanistic aspects from a single tumor, but across tumors, it's still sort of limited. Usually data sets I have seen at least are 5, 10, very double digit or just single digit. So what are some of the analytical challenges? I don't want you to read this slide, but I'll tell you broadly speaking, there are two. One is just the pre-processing and quality control. And especially in imaging, and folks who work in imaging might appreciate this. It so much depends on the type of instrumentation that's used. Saying simply, there's a red color in one image matches the red color in the other image because you're trying to do population statistics. So there's a lot of normalization and standardization that goes into it. For some of the imaging technologies, it's pretty well oiled, but for the newer ones, it's still a work in progress. And then even though you get a reasonable solution to that, the next bucket is, just the modeling challenges, because just to load this data up, we are talking, either thousands or even million or up to, a million cells per sample, and then, ingesting that data and now think about multiple patients, So that's one of the, aspects of it. The other is tumors, when you do especially population analysis, tumors have different shapes and sizes, So imaging, when usually things like brain imaging, it's a very set template, the northwest corner of your brain, of every human brain is sort of similar. That's where so everything is mapped onto a template, but tumors you can't map to a template. It's like, to draw a crude analogy, it's like ginger, right? You pick a piece of ginger and you pick another piece of ginger. One could be more rounded, one could be more speculated. So how do you do analysis for non-conformable spatial domains? That's one of the modeling challenges. And as you might imagine, this is very high resolution data that we are looking at. So high degree of spatial data. And I'll give you a specific example. So we wrote a review paper that came, I think, last year with some of my postdocs and colleagues, that goes into these sort of analytical challenges, especially for cancer imaging data, just from a statistical point of view, but also from a quantitative point of view, for modelers. And my pitch here would be, especially being here in the Alan Turing, is this sort of becomes a perfect playground for conflation of AI and statistical models. And bear with me, this will be my qualification slide, and how they can blend together to solve these kind of problems. as I mentioned, that's the pre-processing and the quantification aspects of it, right? And a lot of this is actually rinse and repeat analysis. And I totally believe that the AI and this. In fact, for spatial multiplex imaging, that's exactly what we are doing, because it's really good at detecting data quality issues. If something is not conforming, then it can spit it out. So it can do rinse and repeat analysis very well and accelerate that. But what it can't do is can't fix a bad experiment design. So that's where the human, and more specifically a statistician or a biologist, has to be in the loop, because that's where when the data was collected. So I'm actually in the midst of a iteration with one of my clinical collaborators on this, because we are trying to use this as a prediction tool for immunotherapy response, and the timing of when these biopsies were taken becomes so important, and the way it was taken. So, so we're going back to the protocol and trying to figure that out before we make, pie in the sky results and claims. And then comes that, so that feeds into the statistical modeling aspects of it, and which I truly believe the AI can augment. But a lot of this is actually process-driven models. So process-driven, I mean biologically informed process. So there's a lot known then, about these that can be quantified into specific models where the parameterization needs. I know I'm being a little vague here, and I'll be specific in a couple of slides about some specific examples for this. But biologically, the fact that, tying up specific hypotheses to parametric or non-parametric complex models, but tying them to a specific sort of hypothesis-driven models and informing them about, the model building and organically coming up, I think that's really helpful. And especially in this case, where the variability is so high, the uncertainty quantification it's a very key issue, And then, of course, any kind of statistical model actually backs that in it. And often the clinicians or scientists I work with, translational scientists, they have actually a lot of knowledge already, and bringing their information into this model building process. That's where Bayesian models can be especially amenable when you combine. As simple as something like, is their confidence level low, medium, or high? As simple as that can be built in, to make it a bit more interpretable scientifically, but more importantly, actually. And then once you have done all that, the end products, which could be either research-based or even a clinical implementation. And that's where I think that AI can absolutely accelerate, which is the, not just, in terms of writing papers and drawing pretty pictures and the writing, but also app development, that's something that can be done, in a very fast manner. I remember coding up my first Chinese app ground up, and now, some of my students show me aspects where they can generate an app within an hour or so, based on that. And that's become our modality to share results with our collaborators, And if you get the pizza analogy, if you know of Domino's Pizza, that's actually headquartered in Ann Arbor, Michigan. That's where I'm going to get my pizza analogy here. All right. So that's my quantification slide. Just going back to the spatial aspect, So there's a lot of canonical and analytical tasks with this data. Once this data comes in, either the imaging based or the sequencing based. And you don't have to read this slide, but broadly speaking, it's some variation, a spatial variation of the problems that we encounter in classical genomics. It's comparing two samples, diseased or not diseased. So it's differential analysis. It's clustering, is it spatially clustered? The clustering got a reinvention with the high dimensional data when gene expression came along. So it's spatial clustering. And then there are aspects of region detections, which is sort of like a similar to spatial detection that comes from disease epidemiological models. So, so as I said, the pre-processing, the AI can automate, and these analytical tasks, that's what AI can augment, combined with careful and rigorous statistical modeling. Or even mathematical modeling for that. Sure. So that was my sort of overarching, you know, setup, and I'll give you a few examples, and at least give you the flavor of the examples. I won't go too much detail into the aspects of it. At least give you an idea. So the 3 examples, I picked one each from each of the buckets. One is histopathology imaging, about tumor immune infiltration, cell-cell interactions, and I'll come to that in a second. And also cell-cell interactions in multiplex imaging, you know, how to quantify that and associate that with clinical outcome data. And then another class of problems, I really like networks, and I worked a lot in gene regulatory networks and genomic networks, and the natural extension of that is in spatial networks and how to, you know, do network estimation for spatial data. That's the gist of it. Hopefully, I'll get time to get to it, or at least I'll give you, you know, a high level. So from the histopathology imaging, so this was a project we started working a couple of years ago in melanoma. So melanoma is a skin cancer, and it's known to be a highly immunogenic cancer. That means it's an immune hot cancer. There's a lot of immune activity that happens in melanoma, you know, and that's one of the primary reasons it actually responds fairly well to treatment, immunotherapy. You know, so it's one of the successes in immunotherapy. So the data set we had here, we had the H&E images, the pathology images, and then it went through an ML-based model, a machine learning-based model, where a pathologist delineated each cell as tumor or infection. So we used that as gold standard and trained a whole model to quantify all the cells as tumor. Right, and then we did it a bit more iteratively than that. So essentially what we have is, this image is now a collection of black and red dots, where the black are the tumor cells and the red are the immune cells. And we have on average around 100,000 cells per biopsy, and we have, I think, 330 biopsies, you know, worth of data, 330 patients, that is, you know. And the good thing is the resource where it came from also has multi-omic data associated and patient outcome data, so we can do a lot of association analysis. No. So the first task, actually, a PhD student of mine and we were looking at it. If you just simply, you know, look at the whole thing, a few things stand out. You know, one is the variability. So there are 2 levels of variability that we can sort of glean here. One is a first order variability, which is just the prevalence of cells. How many red dots, how many black dots, right? That's the first order, just the prevalence. And then the second order is, you know, relative to each other, how they are located, because that's the tumor immune infiltration. So I guess the goal here was to just quantify and, you know, understand this variability, and then, you know, can that lead to some insights which can give us, you know, in the context of immunotherapy, you know, some aspects of tumor infection, a quantification of tumor infection, right? So very simply put, the question here is, can we come up with a probabilistic method that can quantify 3 types of interactions, right? Which keeping it simple here. So this is a low negative interaction. That means, you know, the red and black cells, the tumor immune cells are, you know, far apart from each other, or in quotes, because we're still talking very small distances here in microns. A null interaction would be something that's adjacent to each other, and then a high positive interaction, they're just intermixed with each other, right? No. So, and how to quantify this? Like if you see these images, you know, at different portions of the tumor, you can see that different levels of mixing, right? And there's a little bit of a aspect of, maybe I'll get to the model and then say that. No. So, right. So the setup is, you know, hopefully that's clear enough. So it appears like, you know, when we start looking at it, like this is exactly what at least geospatial and spatial statisticians have been working for decades. It's called a spatial mark point processes. And all that means is a random point cloud in space, right? These locations of the cells are random. And then, you know, the mark comes from the fact that these are, you know the identity. means either it's a red cell, which is an immune cell, or a black cell, right? And this can be binary or multi-category. Binary means two types of cells, multi-category is multiple types of cells. We'll come to that when I talk about multiplexing. So you can actually treat each image as a realization of this process. Now, as I said, this has a rich literature, not just in statistics, but also ecology. So there's actually a very cool model called the hierarchical Strauss models. Some of you might have heard about it. So this is the construction, the likelihood construction, but I'll tell you, it actually encodes a key parameter. It's based on counting processes and Poisson processes. But keeping that aside, it has this key parameter called gamma, which actually captures this level of interaction. And that's the key parameter that tells you if there is a negative interaction, a positive interaction, or no interaction. So yeah, we didn't invent this. This has been around for almost 50 years now. It's used in ecology a lot. Then it turns out this is a very natural model for modeling such data. So what it says is there's an assumption of hierarchy that in cancer evolution, right? What is cancer, right? One cell gone rogue. And then it tells the cell right next to each other to go rogue. And then it tells the cell next to each other to go rogue, right? So there's a natural evolution that happened. We are seeing at one snapshot in time when cancer happened, right? So what it says is tumor cells were there, and conditional on that fact, it recruited the immune cells. So the whole the modeling process is based off of that. So there was a systematic recruitment or a disbursement of these immune cells, right? So that's why it allows actually both positive and negative interactions. And then it's a very naturally sort of a process model for this data that being and provides a generated model as well, because it's got a definite likelihood. And it's actually embarrassingly easy to fit. All you need is, there's something called a Poisson trick, and all one needs is a variation of the logistic regression, essentially, to fit these models. It's got a very complicated likelihood, but there is an approximation, which is a really good approximation. But having done that, that actually turned out to be just half the problem solved in finding the right model. It turns out that actually this interaction parameter that encodes the positive, negative, and zero interaction, you get a number out of it. You run through a process, you spit out a number, 0.1, 0.2, 30, 40, 100, so it's unbounded. The key question is how to determine significance here. And that's a very nuanced question because, to find out how high is high, how low is low, it's very relative to the number of cells, the prevalence of the cells. So it turns out actually it's a cute inferential problem to determine significance here. So we actually, working, putting a Bayesian aspect to this, one can actually compute this, what we call cell type interaction probability. And without going into the mechanics, the broad idea here is that, you get a posterior distribution, and then the prior distribution, the null distribution, is actually computed for each biopsy separately. So the null changes with the biopsy, so essentially. So it's an observed empirical null. And then, looking at that overlap of the two, you get an probability. So it's highly interpretable. It's on probability scales. Close to 0 is low interaction, close to 1 is high interaction. And it can cheaply be completed with MCC samples. But the only caveat is the null distribution has to be calculated for each biopsy. But that can be parallelized across biopsies because it's done one biopsy at a time. Right. So essentially, that's the idea. You strip out the cancer, you take out all the tumor cells here, and then you get these locations, or take the tumor cells, sorry. You remove the immune cells because the null depends on, it's a hierarchy, right? So the tumor cells were there, how much did it do? And then you generate a lot of null sort of random Poisson processes and aggregate those. That's all. The computation happens. So essentially just pulling it all together, briefly here, so you have the each biopsy, then you partition them into small units and then basically compute the probability on each of them, and then it will put it back. This gives you sort of a map of human being. So the darker it is, the higher probability it is, the lighter it is. And then once you get this, you can average it out, get a sort of level at the biopsy. And this was developed by Nate Osher, who was a PhD student along with my colleague Gian. And then you can do this for each biopsy. So we ran this method for the melanoma biopsies. So there were 335 biopsies, as I said, computed the cell type infraction probability. So this was more like a positive sampling check. You see, these are more tilted towards the right. That means there's a higher level of tumor-immune infiltration happening, which is sort of expected. Because melanoma, as I said, it's a highly immunogenic cancer, and you do expect a lot of and what's is there an association of that with some of the clinical outcomes? We actually looked at many types associations of CTEP. One was with genomics, one was with survival, and it turns out actually it is associated with survival. There are other types of interaction functions, but it is not, and it's actually significant even adjusting for stage, age, and other factors, which are highly relative as well. So there's some useful information that it does capture. But it left us with one question: is if you see that we get the CTEP map for the whole biopsy, but we ended up actually collapsing it to one number, right? And we were missing a high degree of spatial variability there. So and as you see here, what shows here is that there is a definite mean variance relationship. That means that the variation of CTEP across these biopsies is it varies a lot, both within and across. And so we wanted to understand this, like what is this variation coming to, right, within a given biopsy. And we said, okay, why don't we flip it around and integrate with molecular data, especially gene expression data, and answer a simple question where, how much of the tumor immune infiltration is driven by the molecular application, which is important from the gene expression. Right. So we see the pathology is like sort of the end of the disease, but there's something happened molecularly. Cancer is a genetic disease. So what was that? So, and we wanted to borrow strength across all biopsies, 335 biopsies to do this modeling. And as I mentioned, these are non-conformable spaces. So you have to think a little bit more carefully because these all can't be registered. So if you load one biopsy, which could have, 10,000 cells, another one could have 1 million cells, they are not all matched up. It's not a regularized sort of matrix that you can just put into a R code or Python, right? So that's where, we built a spatially structured regression model where it's basically a population level model, but just to give you an idea, the gene expression actually comes as a covariate in it. And then each of this component models different things. So one models what is the effect of the gene expression within a given biopsy. So that's casted as a random effect. And then what is the gene expression that affects, at the population level, and then, and then we just run a regular sort of a card process, which is a spatial process, So we're just pulling it all together, and this is the model that Nate developed as part of his PhD dissertation, called Dream Space. So essentially, you can think about you're regressing a whole set of biopsies on a set of gene expression, and then that's where the spatial structure comes in. And then as outputs, you get two things. One is genes that drive the overall biopsy between biopsy heterogeneity, and genes that drive within biopsy heterogeneity, right? So they point to different things. So one points to the impact of the tumor microenvironment, but they could be driving in general, tumor heterogeneity as well. So both within and between. And so what? And we use this to model gene expression data from different kinds of immune classes of cells of immune classes of genes, and then you get different kinds of immune classes. So if you're an immunologist here, you know these. One knows these classes. There is called macrophages, neutrophils, and NK cells. And it turns out actually the neutrophil class, especially in melanoma, little is known about this class, but that's what drives most of this heterogeneity. So that was some of the novel associations, but some are consistent, like NK cells and others. How much time do I have?
Another 5 minutes.
5 minutes? Okay, so maybe I'll just go through quickly the spatial multiplex imaging. It's sort of like a natural build-up from this. So the multiplex, as I said, it's a direct sort of build-up on H&E. So the idea here is that you get raw images, as I said, with different types of cells. But there is a pre-processing steps, which is called the cell phenotyping, is identifying the identity of the cells. And basically the types of cells you get is different types of cells, like you can see it in colors here. And again, the principles are very similar. You have a first order, which is just the prevalence of the cells across biopsies, and a second order, which is more spatial interaction. That's on me, right?
I don't know what's.
Yeah. All right. I'll keep plugging. And then that's sort of the biomarker extraction from these aspects, the spatial biomarker extraction, and then you can do a lot of statistical modeling, prediction, clustering, and so on, right? So the AI can automate this, because this is a canonical problem in this. It's still an open problem. There's no clean solution. We have tried a few, and it seems to work because it's just, so there are, to just give you very, a 10-second. So these are basically images, right? These are pixels. The first task here is actually to delineate a cell. So there is actually that's where AI is really helpful. You can actually delineate, segregate a cell. So you get like cells, and then you get multiple pixels per cell, and then you get a value. And that's the value of the protein expression. And that value has to be said, is it high enough to be called a T cell or not? Is it high enough to be called a B cell or not? Right? So there is a level of customization what requires. And as I've said, before, these aspects need proper statistical models to influence. So essentially the data is of this kind. You have each patient actually, most of these data sets, spatial multiplex imaging, they don't actually get one sample per tumor. You actually get multiple spots in the tumor. So these are called stamps. So they basically stamp the tumor at different places and then get like the images of the cells. And you get different types of cells here, right? There are a lot of modeling issues with this data that a simple plug and play AI type machine can't handle. And the reason is this, there is a lot of technical artifacts that happen in this data, which mostly comes from technical artifacts like shearing of the tumor. So there are a lot of holes and spatial homogeneity that comes in. So basically, if you think about this one right here, the intensity pattern here is basically just you're taking a picture of the glass, right? There's nothing there. There's no tumor material there. So you need a scalable metric that can take into account the inhomogeneity of these holes in this tissue to quantify tumor immune cell-cell interaction. So you're going beyond tumor immune interaction, and the fact that you can't treat each image separately because these are nested within patients. You get multiple images per patient. So that's where the statisticians in the audience you can think random effects here. You're getting subsamples per patient. So we developed this pipeline for doing that. And I'll tell you maybe a high-level summary of how we treat this to quantify interactions. So here what you're seeing here is 3 types of cells. Cytotoxic T, PD-L1 negative tumor cells, and T helper cells. These are 3 different functions, and you can see the relative So what we essentially do is we estimate what is called a spatial intensity surface. There are many ways to do that. The simplest way to is a two-dimensional histogram, or two-dimensional kernel density smoothing, right? You get these. And then, so the idea here is you get two densities, and what you're trying to estimate is, are these densities right on top of each other or not, right? So these are two-dimensional. Do the hills match up? Do the valleys match up, right? Or do the flat surfaces match up? So there's a metric of computing distances between densities and many metrics. One of them is called the Shannon distance, Jensen-Shannon distance. And if it tells you, so the higher it is, it is right on top of each other. The closer they are, closer to zero means they are farther away. And actually, this is really simple idea, but it actually works really well. We've done our simulations and a lot. So to give you an idea, I'll show you here. These are six types of cells and their intensity surfaces shown in 3D. And if you see this one right here, CD19 and CK, so these are tumor cells and these are CD19 cells. So I would expect the distance between them to be, so closer to zero is perfect overlap, one is no overlap, right? So this should be closer to one. And if you see here, the distance between CK and CD19 is yellow, and it's like closer to one. And these two I would expect to be right on top of each other, CD14, CD19, CD14, CD19, you can see it's on the lower end, so it's lower, right? And as I said, like we've done those simulations, this is very robust data artifacts, like data sources. And once you get this, all you have done is reduced each image to a number, right? And you get these for each pairs of cells. So if you have six choose two numbers. So you get these distance matrices, and these can be mined for you. You can put them as a covariate, you can do clustering, you can do, And the one nice way is you can extend to three-way cellular interactions. What that means is, we are seeing pairwise, right? Cell A and B are interacting. But biology is so complex. A cell of type C might be mediating the relationship between A and B, right? So that's three-way. So you can do three-way too. Correct. And we have run this on a host of in-house data sets, and just a small smattering of the results. Response to immunotherapy in lung cancer, the quantification, quantification of cell-cell interactions, how they're impacting response to immunotherapy. And this is my clinical collaborator, real quick. The reason what we are trying to do here, our pipe dream is to develop a predictive model using these multiplex images, because these are cheap to get. But these are cheap to get for a research-rich university, because these are multiplex images that people can. But what we really want to do is come down to a minimum set of cells that will be predictive of immunotherapy, so that this can be deployed in less resourced places. And that's something, it's a long way away. We are trying to do that towards that. But I'll skip this portion, and I have some references here. This will take a bit to unpack. So I'll mention a few things like, for each of this work that I showed, we actually built up GitHub sites and softwares. And especially for the DIMPLE, which is the spatial multiplex imaging, actually there is a Shiny app as well that, one can actually upload their images and work with them. So it's sitting on you can either download it and run it locally, but you can also actually run it right off the Shiny apps or POSIT server now that you can run with the other two, which is the original part of it. So just closing, I'll pick up a few points. Hopefully I've conveyed my, enthusiasm here about spatial biology. It's got immense potential and promise. But again, it has to be grounded that we have to look at some level of clinical translation with this. For discovery, it's very cool modeling. Games to play here, but development of clinical prediction models. That translation impact will be the next step in seeing how useful they are. So this is right now the hype curve of things, but it has to land somewhere to make an actual impact. And that's where I think from a discovery aspect, integrating with other omics, I think people are mapping the whole atlas of the tumors, the 3D of the tumors. Every cell will have an identity. Let's see where that goes, going from 2D to 3D. And of course, my spiel here is uncertainty, variability, configuration is the key in this data. So there's a lot that's happening both from a design perspective as well as modeling perspective. All right, so I'll end there. Hopefully I've convinced some of you, if not confused all of you. Happy to take questions both here and online.
Great, thank you so much. Yes, and thank you, Veera, and we're very happy to receive questions online and in the room. In the room, please just raise your hands. Robin?
Just a really practical question. It was interesting when you said about you could tell there were gaps in the when you're looking at the sort of slides, because essentially, I guess it's how it's been sliced. So, I guess firstly, is it sort of is there then I know this is probably a completely different area, but then is there a particular technique or a better way to take the tumor slice that would allow more information to be or less information to be lost, or is that not really relevant? And then are there sort of gaps that you can definitely tell are there, and are there gaps that are probably there but you wouldn't know? It's kind of like I always view everything with missing data. So it's like there's missing data that you know is there, and missing data that you don't know is there, but could be there, but you just wouldn't know.
Right, that's a really good question. At a very sort of local level, you could do some missing data, right? Like there's one particular data sets we are like, we're dealing with this, we're looking at different points along the tumor, right? And we know that these two are right close to each other. We have the location of the two images. So you could do some missing data imputation to figure out like what was the distribution sort of that. No, so that's, that can be done. I think that's justifiable to some degree. This one, on the other hand, is a little bit different because this just happened as during the part of the instrumentation that they put the pathology slide on the tumor, and they were just to show that they stretched it out. So it's actually, there's no data there, there's no cells there.
Yeah.
To go back, like you have to go back to the raw image to figure this out because it's coming from the background. It's not coming from the actual. So. And sometimes getting that background correct is really important because and that also varies between slides. If it was run on a particular day by a particular person, So, like to go back to be more specific, like these areas, probably like smaller areas, you can do a missing data imputation and get the overall. But for these ones, you need to go back and look at the actual data collection procedure. Yes.
How far in the future do you think that this kind of work with some of the issues that you've identified where more work needs to be done, how far in the future do you think this may have clinical application?
I think we are, of course, like we can come down, like based on the data sets we have, we are still looking at, there are a few things there, I think, to solve that entire, solar spectrum. The first is we are still working on a smaller data set. Like cancer, usually you don't get a big data set like GWAS studies, thousands. This is biopsy samples. So we are still working in the hundreds. So we got to do best what we can validate, right? We can do some validation based on whatever we have. And then hopefully someone else will replicate us, or we get an independent data set, we replicate it. So I'll take a concrete example, response to immunotherapy. So we are working on a prediction model using spatial biomarkers to work response to immunotherapy. But ideally, we could publish that, like it's doing really well. We know exactly what spatial biomarkers are important in that. But I would like to see it validated completely independently. And then coming down to a set of markers, which, you don't need 20 markers. I think the my clinician feels like you can do with 3, 4. Just get me the main ones so that I can get this into a IHC and then we can work out an assay and that can be deployed.
And when you say response to immunotherapy, do you mean in cancer rather than other disease areas where immunotherapy is used?
Yeah, the concept carries forward, right?
Right.
The cancer, you're responding to it, to a particular intervention. So you respond or you don't respond. Or, yeah, so that's one particular set we are working on. It's a few years at least.
Yeah.
Yeah, I would like to see a good validation of it before we sort of build a whole assay based on that.
Thank you.
That's all. And increase our data set size too. It could be a quantitative validation. I'll be happy with the qualitative validation too. That similar sorts of things pops up. Because what we are trying to unravel here is the roles of these different kinds of cells, right? Does
Yeah.
it matter you have too much T cells in your body and working to recruit B cells? That's enough.
Because this would have applicability in conditions like in autoimmune disease, that kind of prediction model.
That's right.
Yeah,
Yeah, anything you can get a tissue sample out of.
yeah, yeah.
Thank you. Any other questions in the room or online? Right.
Hi, thank you for the sharing. May I ask a question?
Sure.
Okay. I'm very curious about the first work. It introduced the interactions between tumor and immune. But my question is, how about the stroma cells? Will that be included or?
Yeah, so that's a really good question. So right now we just focused on pairwise tumor immune, right?
Yeah.
We only looked at the tumor. We didn't look at the stroma in this one, right? But that's a good question. So in the multiplex imaging, maybe I have it here, you know. So actually in the multiplex imaging, we are actually breaking down the interaction into specific to tumor regions and the stromal regions because the interaction might be completely different. No. So we do it separately for each of the regions, the tumor and the stroma, as defined because there is a specific marker for stromal cells and a specific marker for tumor cells. So we can segregate both those regions spatially, and then where am I talking? So, and then we can do quantification of interaction separately and see if it is still there. Or what the differences might be.
Okay, thank you very much.
Thank you, Yipei, for your question. If
Thank you.
we don't have any other questions online, the in-person participants could continue the discussion over some snacks. Thank you very much, Veera. Yes, I will share the slides and the video will be available on the channels. Thank you
Of
very
course,
much.
feel free to contact if you have any questions. Thank you.
Thank you.